Okay, we are live.
Okay. All right. Good afternoon. Let me just put this in presentation mode. Okay, so this is the last day of, can you confirm you're seeing the presentation full screen or you're seeing, let me swap, I don't think so. Now it should be full screen, right?
Not yet. I think it should come up anytime. But I still seeing your slides.
Full screen.
Not.
Can you see two screens or one?
Okay, just one screen.
Okay, so that's
slide.
it. Okay. All right, thank you.
Yeah.
So, as I mentioned before, this is probably the last module 0. And I'd like us to talk about two things this time. Let's hope we can manage it. One is the principles of evolution. And then the second thing that we're going to talk about is the central dogma. So transcription, translation, proteins, and so on. So those of us who are genomic scientists, the reason why we do genomics experiments is to ask certain questions. For example, why is it that 2 E. coli daughter cells look like the daughter cell that's preceded them? Or why do you look like your parents? Okay, but you're not your parents. So why is it that then there are things, features that you have that we cannot find one or both of your parents? How is it that all this information that describes you is packed into your cells? Okay, and this is something that we've talked about, how DNA is a repository of information that the cell can then read or transcribe in order to make things. Okay, and then how does this information determine how the cell works? So yes, the DNA is a repository, but how is the information then unfolded to make enzymes, to make structures, to make a cell do things, to make a cell move? And then what dictates the diversity of species? Why is it that not all bacteria are the same species? Not all E. coli are the same. Not all E. coli subtypes within those subtypes are the same. So one of the things that you have to understand in order to make sense of genomes is you have to understand the basics of evolution. And many of us would have heard, even if you're not familiar, of Charles Darwin, who went to islands in the Galapagos, saw, birds, finches that were genetic siblings, but had very different features based on their environments, and came up with the core principles of evolution. Something that people often don't realize is that the first phylogenetic tree that was drawn from an evolutionary point of view was actually Darwin's. So this is a page of his notebook. He's not drawing it based on data. He's drawing it based on ideas that he has. Now, normally, if you're more than in part 1 in university, normally, you should know about Darwin. The schools
Hello,
have
Prof.
taught that.
Sorry to interrupt you.
Yeah.
We can't see your slides. Like, it's not moving.
What slide are you on?
We are still seeing the first slide.
Okay, let me try and stop sharing and share again.
Okay.
Screen. No, I don't want to share the screen, so let's stop. How do I stop now? I really don't like Google Meet. Gosh. Okay, cancel. It says it doesn't have permission, but it just shared. Share window. Share. Oh, before I select the window, let me try and put it in presentation. Share this window. Presentation mode. Okay. What are you seeing now?
Okay, we are still seeing your PowerPoint. Like it's not on slideshow yet.
So can you see one slide or two slides?
So we can see one slide along with the other ones lined up towards the left.
Okay, so you're seeing my other screen. So let me swap displays. Now what are you seeing?
Same thing.
I don't know why you're saying that. Should I take it out of slideshow?
Okay, maybe that could also work.
Okay. So are you now seeing just a non-slideshow presentation?
Yes, just that.
Okay, let's go with that. I can live without animations and so on. Okay. All right, so I was just saying that normally last year of secondary school, first year of university, everybody should know Darwin, everybody should have read Origin of Species. And it was actually a surprise to me when I came, well, not too much of a surprise because I didn't read Origin of Species when I was an undergrad either. And when I came, I was asking my students about reading Origin of Species. Most of them said that they were never told to read it. They were never actually taught evolution. I know in my own, as an undergrad, I was not taught evolution, but that was because of a shortened semester. So I just assumed that, so because the semester was shortened, we didn't finish the curriculum, and so I wasn't taught. And later on, I was forced to, read up on my own. But later on, in interacting with colleagues, I realized that many of us are actually scared to teach evolution because we think of if you teach evolution, you might change your students' beliefs. And I just had this conversation with the Part 5 students about a week ago. This statement on the next slide, nothing in biology makes sense except in the light of evolution, is a statement that was made by an evolutionary biologist known as Dobzhansky, who was also a priest. So some people think those two things are incongruous, that, oh, if you believe that, the world was created by a supreme being, then you also cannot believe in evolution. Now, this is not the case. Most of us will accept that, yes, Abib.
So that was a mistake.
Okay. All right. I think the advantage of not having it in presentation mode is I can actually see the hands. So most of us will accept that in the days before Alexander Fleming, resistant bacteria were few and far between, and now they're very common. So the bacteria have changed, they have evolved. And whether you believe that Adam and Eve were created and put down on earth, or that they evolved from some other pre-human species, the fact of the matter is that in your lifetime, bacteria have changed. If I grow up a culture today, and we'll talk about some of those experiments, of sensitive bacteria, and plate that culture onto plates containing streptomycin, I see a resistant organism. That is evolution. And that's what we mean when we say nothing in biology makes sense in the light of evolution. Yes, there are aspects of evolutionary theory that ask where life came from, where we came from, and so on. But you don't necessarily need to believe those aspects to understand the fundamentals of how evolutionary theory works. And one of the things that I like to tell students, just to keep their minds at rest, is that there's a fairy tale known as the Water Babies. By the way, has anybody, there are not many people on today, has anybody read the Water Babies before by Kingsley? It's a fairy tale or been told it as a child. I'm sure it's in Kennedy K Library. Actually, it's free online. Okay, so if you like fairy tales, it's a nice one. It's one of those fairy tales that goes on and on and on. Okay, but at one point, Tom, who is a water baby, so he looks like a human being except that he lives underwater. He's swimming underwater, and his quest is to find Mother Nature. And so he goes on this long journey asking, who knows where to find Mother Nature? Who knows where to find Mother Nature? And then in the end, he finds Mother Nature. And when he finds her, she's sitting like a stone statue. She looks like a stone statue. She doesn't move, although she talks and he hears her. So he said, you can't be Mother Nature. He said, why? She said, why not? I'm Mother Nature. Everybody knows I'm Mother Nature. He said, no, I expected to see Mother Nature sowing flowers, molding animals, making things, because that's what Mother Nature does. And she said, any fairy or any goblin can make flowers and animals and so on. You just have to be taught, and then you know how to do it. But I am Mother Nature. I make things make themselves. So when you're thinking about evolution, it doesn't necessarily mean that you don't believe in a creator. Okay, I'm not going to go into the details of, what is the actual, because in actual fact, we don't know. We either work on evidence or we work on faith. But I want to assure you that you need to understand evolutionary theory in order to understand how genomes change and why it's significant. So what's evolutionary theory in a very short nutshell? It's one of the 5 great ideas of biology, some of which you may have noticed on some of the slides. The first is that the basic unit of life is the cell. This I'm sure we all know. And the basic unit of heredity is the gene. We've talked about that. Okay? And then evolution by natural selection, which we're going to talk about in a minute, is a characteristic of life. And then everything in life is based on chemistry. So you and I are made up of chemicals. DNA, we saw, is made up of molecules. Okay? And then biological organization is based on logical, informational processes and structures. Okay? So how does evolution actually work? The way evolution works is that existing genomes change. Okay? There is variation. So what you're seeing on this slide, not in his exact words, but this is what origin of species is about. There's variation in life, and that variation can be inherited. Okay? And then there are environmental conditions that make some variants more successful than other variants, such that there is differential reproduction. Some variants will reproduce more than other variants, and so they will leave down more descendants. Okay, now let me just correct a few misreadings of evolutionary theory. We've corrected one of them. That is, understanding evolutionary theory does not mean that you're a heretic or an atheist. Okay? Another one is not that a living thing sees a characteristic that it wants to do, or that it needs, and then it evolves towards that. That is Lamarckism. That was a theory that existed before Darwin. Okay, that the giraffe needed to eat the leaves on top of the tree, so he stretched his neck and his neck became longer. That is not how evolution works. The way evolution works is that there's always variation. And then some variants are more successful than other variants. And those variants that are more successful, what I mean by success in this context is leaving descendants. Okay? So, for example, someone might say that I am very successful. I'm a professor. However, from an evolutionary point of view, I am not, because I have only left one descendant. I've only had one daughter, right? So success from an evolutionary point of view, the rats that live in wherever it is that you find rats, because they've had 20, 30 baby rats, they are more successful than you, okay? When we say survival of the fittest, the word fit in evolutionary theory means living descendants. So it's not, oh, I'm fitter than she is because I go to the gym and she doesn't go to the gym. It is that whoever is fitter leaves more descendants. Okay? Now, evolutionary theory, before the time of Darwin, this was a very novel idea. After Darwin, there was just so much evidence that it had to be accepted in biological circles. However, the resistance that came to accepting evolution was the same as the resistance I described at the beginning of this discussion that we have here, which is colleagues, biologists, being cautious about teaching evolution because they are presuming that that would change people's beliefs. But in actual fact, the evolution that is most relevant to pathogens, microbiology, and so on, this was the last area in which evolutionary theory was accepted. And it's the one, paradoxically, that we see. For example, in the year 2017, SARS-CoV-2 did not exist. And in the year 2020, it suddenly appeared in China. And then by the end of 2020, it had gone all around the world. Okay, that's an evolutionary paradigm that all of us witnessed. And so microbial evolution in particular is very accessible because all of us see it happening. Okay, but it was controversial. And controversial because microbiologists could not actually figure out how it worked. So there were two important experiments that made us understand evolution in microbes, and that then made us reinterpret what Darwin had done with Origin of Species and be able to use it to understand how microbes, and in particular pathogens, change. Okay? So the first of these experiments is an experiment known as the replica plating test. And it was done by Lederberg and Lederberg. They were a husband and wife team. Okay? So here's what first of all, let me just set the scenario that they were working with. If I grow up a tube of E. coli and grow up an E. coli culture overnight of E. coli that are sensitive to streptomycin, if I take that overnight culture and plate it on nutrient agar containing streptomycin, invariably, sometimes I don't, but most of the time, I'll find a few colonies growing. So this is I started with bacteria that were sensitive to streptomycin. I grew them up overnight. I plated them out on media containing streptomycin. And then maybe I saw one colony, maybe I saw 10 colonies, maybe I saw 6 colonies, and so on. This happens all the time. Microbiologists see it. And so the question became, where did these colonies come from? If you look at these colonies, you find that they are very, very, very similar to the bacteria that you started with. In fact, they share all the properties with the bacterium that you started with, except that they are streptomycin resistant and you started with the streptomycin sensitive. And if you subculture them, their descendants are streptomycin resistant. So they pass that on. It is something that's actually inherited. So there were two hypotheses around this. One hypothesis was that there's something about putting bacteria down on streptomycin that makes the bacteria change and become resistant. The other hypothesis is that bacteria change all the time. to all sorts of different things, including becoming streptomycin resistant. And so in that my culture, there were a few bacteria that had changed to become streptomycin resistant. And when I put them on a streptomycin plate, all the sensitive bacteria died, and the few that were resistant survived. So these two hypotheses were out there to explain why it is that the bacteria on the plate, the few colonies on the plate, were actually resistant. So do we understand the two hypotheses?
Yes, ma'am.
Okay, thank you. So here is how Lederberg and Lederberg tested the hypothesis. They grew up the culture, okay, and then they plated it on a plate without streptomycin. So sensitive bacteria plated on a plate with no streptomycin, okay? And then they made a block of wood that was the same size as the, let me put on my camera and just show you this. It was the same size of the plate. Let me stop sharing so that you get a big window. Okay. So they made a wooden block, something like this. And then they put a piece of velvet material, not tissue. I'm using tissue just for illustration, okay? So they put a piece of velvet material. The velvet is sterile, so there are no bacteria on the velvet, okay? And then they're going to open that plate that has a lawn of bacteria growing on it, and they're going to use this block to print on the plate, and then print onto another plate with no streptomycin and a plate with streptomycin. Since it's printing, you know, hopefully we know what velvet is. These days, we use it to make scarves for our hair. It looks like carpet a bit, okay? So we know that when we touch the plate, it's going to pick up bacteria on the plate. And it's going to be able to transfer those bacteria in the same position to another plate. So we're going to, from the original plate with no streptomycin, we're going to print two plates, one with streptomycin and one without streptomycin, okay? So when we do that, we have two duplicate plates. The only difference between them is that one has streptomycin and one doesn't. And we put them in the incubator. The next day, what happens? The one without streptomycin, we see a lot of bacteria growing on it, as you'd expect. The one with streptomycin, most of the bacteria cannot grow because they're sensitive. But we find a few bacteria that are growing, as we always do when we plate from sensitive culture to resistant. Okay? Now, because we have this duplicate plate that contains bacteria that have never seen streptomycin, we can go to the same position on this plate, and ask, is this colony growing right here? Is it streptomycin resistant or not? Okay? And when they transferred, they found it was streptomycin resistant. Transfer any of these, they're sensitive. So the same position where you find a streptomycin resistant colony in the streptomycin plate, at that position, you will also find a streptomycin resistant colony in a plate that has no streptomycin. What does this tell us? It's not the streptomycin that is causing the bacteria to become resistant. That one variant evolved sometime from the time I picked my sensitive colony and grew it until the time I plated. And that one colony is streptomycin resistant whether I plate it on streptomycin or not. Now, if I plate it on no streptomycin, it actually competes with all the other bacteria for nutrients. Okay? It might be neutral or it might even be slower growing. But either way, it's in the minority. On the other hand, when I plate it on streptomycin, nothing is competing with it. So it has a selective advantage, but it only has that selective advantage in the presence of streptomycin. Any questions? Okay, I don't see any hands. And where's our chat? Okay, I didn't see anything in the chat. Oh, there's a chat right there. Imo had asked, are we taking questions from previous classes? I had actually said we should start that way, but question came in a bit late. So let's continue. When we get to the end of evolution, we'll take questions before we talk about transcription. Okay? All right, the second experiment that explained how evolutionary theory works so well in bacteria is an experiment known as the fluctuation test. And it's an experiment that was done by Luria and Delbruck. So their scenario is similar to that of Lederberg and Lederberg, except that they're not working with an antibiotic, okay? Lederberg and Lederberg, by the way, they were working in the 1950s, okay? If you remember, Alexander Fleming was 1928. So antibiotics had been discovered by the time they were working. But Luria and Delbruck were actually working right before Alexander Fleming. So they didn't have the luxury of being able to work with antibiotics. But they had bacteriophage, okay? And what they found was that if you take E. coli that are susceptible to bacteriophage, when you infect them, they die. But every now and then you have an E. coli culture that you infect with bacteriophage, they're susceptible, most of them die. And then you get one colony that's resistant. By the way, the phage they're working with are lytic phage. That is phage that will always kill. They're not lysogenic. Some of you may have done lysogeny where the phage lives inside and so on. In this case, T1 is lytic. You get T1, you die. If T1 can't kill you, then you are resistant. Okay? So they notice that every now and then, there are bacteria that are sensitive. You infect them with T1 phage, and they are survivors, and the descendants of those survivors remain resistant. So you see that the scenario is exactly the same as Lederberg and Lederberg. However, they're using bacteriophage instead of streptomycin. Now, what's interesting is that although the scenario and the question was the same, the way they approached the question was completely different. Okay? So, Luria and Delbruck felt that if the bacteriophage were causing the bacteria to become resistant, then that would mean that if you had, say, a liter of culture, and you split that liter up into 100 ml aliquots, each of those aliquots should give you the same result on exposure to the bacteriophage. But if the bacteria just change randomly to, we now know that they mutate too, but in those days that concept was not understood. If the bacteria just change by chance, that means that if I have individual cultures that make 1 liter, okay, say I have 100 10 mil cultures, each culture will give me a different result. There will be fluctuation. So they did the experiment. They did a big tube, and then they did little tubes. And if the phage were causing the bacteria to be resistant, we should see the results on top with the little tubes. But if the phage were not causing the bacteria to be resistant, but the resistant bacteria pre-existed, then in some cultures there will be many pre-existing bacteria. In other cultures there will be few. And this is what they saw. They saw this fluctuation. And based on this fluctuation, they concluded that the resistant mutants were there before seeing the bacterial phage. Okay? I will send you Lederberg and Lederberg's paper. It's very short. It's easy to read. You can find Luria and Delbach's paper online. It's also fun to read, but it's a lot longer. But I will send you a short paper on it, or a kind of textbook excerpt on it, so that you can further delve into the concept. Okay? The fluctuation test is actually very clever. In fact, now that we know that what's causing it is mutation and so on, we can actually use the fluctuation test to calculate mutation rates, okay, which is also a very exciting area of microbiology, but one that's beyond the scope of bioinfo UI. It's actually microbiology, not bioinformatics. So we know now that Darwin's principles operate in full in microbes. The organisms change. When they change, there are variants, and under different conditions, some variants may have a selective advantage. Okay, I'll talk more about this in a bit. I did, however, want to mention that the rate at which different organisms change varies. It varies within organisms because there are certain chemicals and physical conditions that can cause DNA to change. These are DNA damaging agents or mutagens. So if I expose bacteria to UV light, I get many more streptomycin variants in Lederberg and Lederberg's experiment than if I don't, because UV light is a mutagen. But if I don't expose the bacteria to UV light, I still get change, because change is normal. It's a facet of life. Okay? And we mentioned before that the major reason for normal change is that during replication, there's a small chance of error. That chance of error comes down due to proofreading and also due to repair, but it's always still there. Okay? Now, if you look at organisms across the tree of life, okay, multicellular organisms have the lowest mutation rates. Okay, they actually have more repair systems. I don't like the slide because it refers to some things as higher eukaryotes. Believe it or not, there's nothing like higher and lower. Everything is an organism. Okay, but anyway, eukaryotes and multicellular organisms in particular tend to have very low mutation rates. Okay, and we'll talk about why in a minute. Bacteria, low mutation rate, but slightly higher than multicellular organisms. And then viruses have much lower, higher mutation rates. Higher mutation rates, lower fidelity of replication. Okay, and we mentioned before that the RNA viruses have the highest mutation rates. Okay, now why would eukaryotes have very low mutation rates? It turns out that the majority of mutations either have no consequence or a really bad consequence. The type of mutation that either doesn't kill the organism or has no consequence at all, that's rare. This is because, first of all, not all DNA is coding. So if you have a random mutation during replication, and that mutation occurs in a non-coding sequence, then it may not lead to any effect. It may, but it may not. Okay. Secondly, if that mutation occurs in okay, here we are. If I have a mutation in a codon, this is encoding sequence that changes a UUU to a UUC, there's no effect because I'm still getting a phenylalanine in that position. And we talked about the fact that even if the amino acid is changed, if it's changed in a part of the protein where it's not required for function, then the function may not change. Okay? Now, that's one side of it. The other side of it is that when there is a change, it's more likely that that change will be deleterious than useful. Okay? This is because we have to think of evolution not as a creator, but as a tinkerer. Okay? So imagine that somebody flies over UI, the UI campus, with a helicopter. And from the helicopter, this is an evil person, okay? So from the helicopter, the person drops a big rock. The person is not throwing the rock at someone. In fact, the person could be blindfolded. Just throws a rock out of the helicopter and then the helicopter flies away. I'm sure you understand that the most likely outcome is that all of us will say, what's that, what's that? But there will be nobody hurt. Okay? That's the most likely outcome. However, it's just as possible that that rock will land on a student coming out of PHM 400 whatever test and hurt the person, right? Now, a few months ago, I fell down and I broke my finger. It was actually bent, and they had to bend it back. The surgeons had to bend it back into shape. You can imagine that if that was the day that the helicopter flew over UI and the rock was thrown out, the most unlikely event of the three that I've described is that that rock would have hit my bent finger and bent it back into shape. So the first one, no consequence. The rock fell, hugely damaging, potentially damaging event. The rock fell, there was no consequence. Or the rock fell and there was a terrible consequence. Somebody was hurt or even killed. And then the third thing, the rock actually did something useful. The least chance you're going to have is the rock doing something useful. This is how evolution works. It's entirely a chance thing. Okay? So after the change happens, that is when the environmental situation will determine what had a selective advantage. Okay, the example we're looking at on the slide. There are beetles. Let's assume that the ancestral beetle is green. Okay, maybe they used to live in the grass. And then there was variation. Some beetles became brown. They had a change in their DNA and they became brown. The green beetles reproduced and had green descendants. The brown beetles reproduced and had brown descendants. And then the beetles started living on a tree trunk. It turns out that the birds that eat beetles, they find it difficult to see the brown beetles on a tree trunk, but they find it easy to see the green beetles. So they eat off the green beetles. In this particular scenario, the brown beetles have a selective advantage. Okay? And then we will say, oh, that this species was originally green. It evolved to be brown. It evolved due to, number one, genetic change, variation. That variation was passed on, heredity. And then it was, the variation was successful in this particular context. I'm sure you can imagine that this same variation, if it had played out on a football lawn, the bird wouldn't have seen the green, it would have seen the brown, and we'll see a different outcome. Okay? Any questions? Look in the chat. Okay, nothing. All right. So I wanted to give an example of how we can actually use this evolution in the lab, in biology. Okay? We've got a lot of people who are either microbiologists or microbiology students, or who are studying microbiology as part of their course. Okay? So most of you understand that if you want to identify a bacterium, what you typically do is you grow the bacteria up to pure culture, you do a gram stain, look at it under the microscope, find out whether it's gram positive or gram negative, and then what shape is it, what's the arrangement, and then you do a suite of biochemical tests. Which sugars does it ferment? Does it produce acid alone or acid and gas? Can it use citrate as the sole carbon source? Can it convert tryptophan to indole? And so many other things. When you've done your battery of tests, you now look it up in a manual like Bergey's manual or something like that, and you'll be able to say, oh, this is E. coli, this is Enterobacter cloacae, this is Staphylococcus aureus. Okay, this is the way that we traditionally identify bacteria. In human health, it's actually very efficient because we have good algorithms. We know which tests to do and good tables. Bergey's manual does very well for clinically significant bacteria. For those of you who are environmental biologists, you probably have to do a lot more work, more tests, more esoteric tests to be able to get an identification. Okay? Now, not too many years ago, the person who discovered the third kingdom of life, who knows who that is? You know, there are no longer two kingdoms of life. We don't say prokaryotes and eukaryotes anymore. We say eukaryotes. We say bacteria or eubacteria, that's true bacteria. And what's the third one?
Active,
Pardon?
active bacteria.
This is Abib. Abib, you are an instructor. You're not supposed to be answering all the questions. Hmm? What did Abib say? Have we heard of this third kingdom? Have we heard of Halobacterium as a genus? Salt-loving organism, Halobacterium.
Yes, we've heard of Halobacterium.
Okay, we've heard of Archaebacteria. Okay, so what Carl Woese was able to show was that the Archaebacteria were more similar to the eukaryotes than to the eubacteria, the true bacteria. Even though they look like bacteria, Halobacterium is a gram-positive rod-shaped bacterium. It looks like bacillus under the microscope. Okay, but it turns out that it's more similar to Plasmodium than it is to E. coli or even to bacillus. Okay, and he was able to do this by sequencing ribosomal genes. Okay, he used the 5S ribosomal subunits. By the way, we know ribosomes and we know they have subunits, right?
Yes, ma'am.
Okay, thank you. So the ribosomes are made up of subunits, and he sequenced the smallest ribosomal RNA, 5S, and from the sequence, from comparing sequences, he was able to tell that archaebacteria were more similar to eukaryotes than they were to bacteria. Okay. Now it turns out that because 5S ribosomal RNA is small, it doesn't have as much information. So we don't use it much anymore. We use the 16S of bacteria and the 18S of eukaryotes to be able to get taxonomic information. And here's how it works. The ribosome is a huge structure in the cell. It's made up of multiple RNA molecules and multiple proteins. Some parts of the RNA molecule are required for the ribosome to function. Okay? Those parts are what we refer to as conserved. So if I take out the ribosomal molecule and I stretch it, ribosomal 16S ribosomal RNA gene, there are certain parts of it that do not vary between organisms. They have low levels of variability. And there are other parts that have high levels of variability. The parts with low level of variability are the parts that are needed for function. Whereas the parts with high level of variability are not needed for function at all. Because the way the ribosome is structured, some parts are just offering reinforcement. So any nucleotide could be there, it just needs to stand. Whereas there are other parts that are catalytic. They need to catalyze something. So it matters which residues are there. So the ones with low residues are where it matters. Low variability is where it matters. And the ones with high variability, it doesn't matter at all. But remember that every bacterium has a 16S ribosomal RNA gene because every bacterium has a ribosome. So we can look at the sequences in the variable regions, and they'll be different from one species to another. For species that are closely related, like E. coli and Salmonella, the variable regions will not be the same, but they will be similar. For species that are very different from each other, like E. coli and Staphylococcus aureus, the variable regions will be very different. So we can actually use now the 16S gene and the information in its variable regions to determine which species we have. And conveniently, just close to the beginning and close to the end, there are regions of low variability, which means that I can actually put PCR primers in regions of low variability, amplify regions of high variability, and then sequence them. This is a perfect alternative. Just a minute. I think the part 4 test has ended because there are some excited people outside my window. And they are blocking us from here. Please, I'm having a class. I'm having a class. I'm teaching a class. It's noisy. Thank you. Okay. Apologies. So we can use the information in the variable regions to actually name the bacteria if we have a database of sequences. And we do. So nowadays, if I have an organism and I can't identify it biochemically, I just amplify the gene and then I put the sequence in the database as a database on NCBI that at some point you'll learn to use when you learn to use BLAST. And then I can tell you the name of the bacterium. Very, very powerful. An example of how sequence-based work allows us to do something that we're doing before, but do it more efficiently. Okay? So actually, if you look at taxonomy now, a number of species that have been described on Earth. Okay, Carlinia started this. Number of species described on Earth in the 1900s was very, very small. Okay, and by the 1980s had not increased significantly until we started sequencing 16S genes. Okay, and now there are thousands. This is not 100. This is something like 100,000, I think. This is thousands of species. But now there are millions of species that have been described because we can use 16S or 18S if you're working with eukaryotes genes to be able to identify them. Okay, any questions? I can see Abi puts Kawus in the chat. That's the person who figured out the third domain of life, but also figured out that we can use ribosomal genes for taxonomy. Okay, I don't see any questions. So let's go on to talk about transcription. But before I want to talk about transcription, I just want to cement some of the things that we've said about DNA and errors and mutations. Okay. We saw this slide earlier, I think our first lecture or our second lecture, where we saw that there are errors. The error rate is extremely low. But it's low enough, if you think about it, now that you think about it in the context of understanding evolution, the error rate is low enough to ensure that we don't end life on Earth, but high enough to ensure that we have the variation that we need to continue life on Earth. Okay, it's actually quite fascinating to think about. Okay, and when you have well, let's skip this because we've talked about it. When you have changes, we mentioned that there are different types of changes. Sometimes you can have a nucleotide substituted. Sometimes you can have an insertion. Sometimes you can have a deletion. Sometimes you can have multiple nucleotides inserted, multiple deleted. And we mentioned the fact that those that produce frame shifts, changes in the reading frame, are more deleterious than those that just change out a nucleotide. And even changing out a nucleotide, we talked about how changing a nucleotide at the first position in a codon is usually much more problematic than in the third position. Okay, we change TAA to TAG. That is changing a stop to a stop. Nothing happens there. Okay. Now, when DNA is being transcribed to RNA, the process is analogous, not the same, but analogous to replication of DNA. Except the enzyme is not DNA polymerase. The enzyme is RNA polymerase. RNA polymerase is forming the phosphodiester bonds between different nucleotides in RNA. Okay? And it's using the DNA as a template. And remember that when we were replicating, the nucleotides were coming in on their own accord by hydrogen bonding. It's not RNA polymerase selecting which base should come. Hydrogen bonding is doing the selection. So the green is the DNA template strand, the orange is the RNA strand, and hydrogen bonding is selecting which nucleotide comes, and RNA polymerase is just making the bond between the phosphate and the sugar. Okay. Now, one of the things to recognize is that many genes are transcribed at once. And in fact, a particular gene may be transcribed over and over again the same time. However, there are important cues in the DNA that determine where transcription will start and where it will stop. RNA polymerase does not just fall on a random piece of DNA and start transcribing from anywhere. And at the same time, when the RNA is made, the RNA gets to the ribosome. The ribosome doesn't just start translating from anywhere. If it did, there would be errors because remember, they are reading frames. Okay? That notwithstanding, there are errors in transcription, and the errors in transcription are much higher. The error rate in transcription is much higher than the error rate in replication. But that doesn't matter because in transcription, we're making many molecules from the same template. Okay? So look at it this way. When you're transcribing, you're just making a copy. When you're replicating, you're actually copying the original information. Okay? If I want to have a service in church, and I have the program for the service with me, okay? The original program that I deliver, if it is correct, and in the process of photocopying it, occasional errors are made, that means that most people in the service will have a correct copy, and a few people will have erroneous copies, and they will just share with their neighbor. But if I bring in a copy that has an error, then everybody in the church has a wrong copy. So for this reason, errors in transcription have less of a problem on the organism. Okay? Now, how does transcription work? RNA polymerase has many subunits. The subunits that do the transcription are called the beta and the alpha subunits. Okay? However, they cannot on their own accord land on DNA, separate the strands, and start transcribing. They need to be brought by the sigma subunits. The sigma subunit is what finds where to start transcribing and recruits the other subunits. Open the DNA duplex unwinds, and then transcription can start. And once transcription starts, actually sigma is not needed anymore. Sigma can leave, which it often does because sigma is often in short supply in the cell. So alpha and beta, they're floating around ready to transcribe anything. And then sigma is determining how much transcription is being done, because it is sigma that recruits, it's almost like the agent, it recruits the other subunits to do the transcription. So how is it that in this illustration, you'll see that sigma started transcription at the red shaded DNA, okay? Bear in mind that in the cell, certain parts of DNA are not shaded red. How does sigma know where to start transcribing? First of all, sigma will start transcribing upstream of a gene or genes. So somehow or other, sigma knows that the gene or gene starts, usually this arrow indicates the start of the gene or something like that. So sigma will always bind upstream of that. And the place where it binds is referred to as the promoter. Okay? Now, how does sigma find a promoter? It turns out that sigma recognizes certain nucleotides in the DNA and binds to them. Okay? So think of sigma as having 2 feet. One of the feet is at the -10 position. The other foot is at the -35 position. The start of transcription is going to be at position +1. And +1 is ahead of the start codon, the methionine that's going to start translation. Okay? So sigma will put 1 foot down if it finds the nucleotides that its shoe binds to. So in this illustration, we're assuming that sigma will see a TATAAT, and when it sees that, it will bind. The other foot will see TTGACA and bind. These are not codons, because these things that sigma is binding to are never translated. They're upstream of the sequence that's translated. Okay? Now it turns out that these sequences that sigma is binding to, you would almost never find a gene that at -10, this would be the exact sequence, and at -35, this would be the exact sequence. What you usually see is that you have something similar to this, maybe TAGGAT at -10 and ACGACA at -35. So sigma does not really bind very strongly. It binds weakly because it finds something similar to its footprints. You'll also see that there are other parts of sigma that look for other things. For example, if you have TG and anything, there's a portion of sigma that will bind to that. There's also a portion of sequence known as the up element, which actually sigma doesn't bind to, the A subunit can bind to. So overall, how well sigma is going to sit, or how well RNA polymerase sits, depends on how good the match is here, 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 and here, and whether anything else is helping sigma to bind. So sometimes we refer to something as a good promoter because the site here is very similar to the sequence here. Site here, very similar. There's a site in between that is TGN, and there's an up element. That's a strong promoter that sigma would like to sit on. But there are weak promoters. Sometimes there's a -10 and there's no -35 at all. Sometimes there's a -10 that's weak, a -35 that's weak, and nothing here. Sometimes you can't find anything. And yet, you know, maybe from experimental evidence that this gene is transcribed. It turns out that there are other proteins in the cell that help RNA polymerase bind to weak promoters. So those proteins, they have their own binding site. They bind to their binding site, and then they recruit RNA polymerase the same way sigma does, and then they can allow transcription to start. So the fact that you don't see all these nucleotides upstream of a start sequence doesn't mean that this gene isn't transcribed. It just means that you, the person looking, do not understand how this gene can be transcribed. I'm sure you'll also understand that it's now very difficult to find promoters, much easier to find coding sequence. Okay, we looked and we saw that when you see a stop codon here, you see an open reading frame, it could be coding sequence. But it's very difficult to find if something is a promoter. Any questions? Okay, I don't see anything in the chat. I don't see any hands up. Are there more people now? Okay. So, in a eukaryote.
Sorry to interrupt you, now. Please, can I go ahead and ask my question now?
Ah, yes, because we're supposed to ask before transcription. Okay, go ahead.
Okay, thank you very much, Ma. Good evening, Ma, and good evening, everybody. So my question actually has to do with something you just talked about now. You have talked about it in other classes. First is whether the regulatory region, that's the promoter part of the gene, only contains non-coding sequences.
Yes. Promoter is
Oh,
always non-coding.
okay, ma'am. Then you spoke about the fact that in a series of codon, if there's a change in the first and the third, in the first is usually, it could be, is mostly has more serious consequence as opposed to the third position. But you didn't say anything about a change in the second position. Is it that there are no changes in the second position?
No, they can be changed anywhere. So what you need to do is you need to just sit down. You know, on the fridge in my house, we have the times table. We put it there when my daughter was in, is it primary 2 or so that every time she went to the fridge to take something, she will see times table. Do the same thing with the genetic code. Not because
Okay,
you're not memorizing the genetic code. When you familiarize yourself with the code, you see some interesting things. Okay? If you look at the genetic code, if you sit down and manually change the third codon each time and calculate how many times it changes the nucleotide, the amino acid, you'll see that that is much greater than the first and that the second is in between. Okay, so if I change AC, oh, let me not use that one. Let me use what's it called now? Okay, let's use stop. If I change, first of all the stop codons, the first nucleotide is a U, okay, or T if you're looking at DNA, okay? So if I change the first nucleotide of a stop codon, I have transformed that protein. Think about it. The stop codon is the last codon in the sequence. If I change the stop codon, what happens? It keeps reading. The protein becomes longer. So actually, changes in stop codons are quite radical. They make the protein longer. Okay? So if I change the first nucleotide in a codon that encodes a stop codon, that is, I change the U to anything, I have removed stop and I put in an amino acid. Longer protein. Now, if I change the third position, if my stop codon was UAG, okay, and I change it to UAA, I still get to stop. There's no consequence. But if I change it to UAC, I get a tyrosine. Okay? Now, if you talk about the second position, I have one chance with the second position if my second position is an A. If I change it from UAG to UGA, I still have a stop. So I still have a chance with the second position to get a stop. With the first position, any change, I lose a stop. And with the third position, I have multiple chances. So you can see just by what I was telling you, you may not actually find it written in a textbook, but it's something that you get by observing the genetic code, although you'll find it in Phil Menini's article of the beautiful intricacies of the genetic code. So read that article, look at the genetic code, and then you understand why I was talking about the third position, the second position, and so on. Phil Menini goes on to speculate that, well, maybe at one point there was a 2 nucleotide code, and that's why the third one is the one that is more more dispensable in terms of change, but that's, he admits, is a speculation. Okay, does that make sense?
Yes, ma'am. Thank you very much.
All right, were there any other questions?
Sorry, ma'am. One more, please. That will also mean that the terminating part of the gene is also non-coding.
You mean after the stop codon?
Yes, ma'am. The part that you refer to as the endpoint.
Yes. So interestingly, after the stop codon, sometimes there's something called a transcriptional terminator. And you know what a transcriptional terminator is? They don't actually look like each other. So the promoter, you could see that there were certain sequences that were conserved. But the terminators, you tend not to find conserved sequences between them. But what you find about them is that they can often form 3-dimensional structures. So tRNAs are an example of a nucleotide string that can form a 3-dimensional structure like a clover leaf, right? And they have to do that for their function. But terminators form a 3-dimensional structure because they actually serve as a roadblock. So as RNA polymerase is coming along, the terminator folds in such a way that it blocks it, and then RNA polymerase cannot continue, and then it falls off. And all of that is non-coding. Okay. Okay, any other questions? Because I did promise that if you have questions from the previous 2 classes, since this is our last session, if you have questions, you could bring them. So did anybody bring questions from previous sessions? Thank you, Emma, for those ones.
Sorry, one more question, please. This is the last one, actually.
Okay.
So the in the example of the Plasmodium novelli gene that you showed us in the last class, the FAT4 gene, we saw that the long stretch of the gene was on different reading frames.
Yes.
However, the FAT5 was just on one reading frame.
Yeah.
I I was just wondering, it being on different reading frames, does it mean anything?
No, it
Is
doesn't
there a
matter.
reason why? Is there a reason why
It
it's
doesn't
not
matter.
on?
No. Well, the
Okay.
reason is that your intron is not in a multiple of 3. So you know the intron is going to be removed. If the intron is in a if the size of the intron is a multiple of 3 nucleotides, they'll be on the same reading frame. If the size of the so if the intron were 21 or 33 nucleotides long, then the 2 exons on either side of it will be on the same reading frame. But if the intron were 35 nucleotides long, it's not a multiple of 3, then when it's removed, the next part of the exon will be on a different reading frame because what was taken out is not in a multiple of 3. So it doesn't matter. And the reason why it's the case is that it's the length of the intron that's between the exons.
Okay, Ma, thank you very much. It's clear now.
All right, you're welcome. Any other questions on previous material? Any questions on today's material? So by the way, this will be our last, we're not quite done, but this will be our last module 0. And then you start your module 1 with the bioinfo team. If questions come up on what we covered in module 0, they can probably answer them. Okay, they sat in on the whole of module 0 last time. They teach module 1, so they have the foundation. So if you have future questions, you can certainly throw them at them. But the whole point of doing module 0 is so that we don't slow down module 1 so much that every single thing they mention, someone has a question. So if you missed any of the sessions before, or if you were late for this one, many people were late, please can you watch back, can you revise and so on, so that we have in the class a reasonable amount of understanding of foundational concepts, and then we'll be able to move fast with module 1. Okay, and then, you know, I'm a professor here. It's my job to answer questions. So if you need to email me anything after today, happy to either respond by email or set up a meeting so that we can chat about something you're not clear about. Okay. There's a question from Joy. During elongation, is it possible for something to cause RNA polymerase to undergo an irreversible pause before reaching the end of the gene? The answer is yes. For example, if a particular nucleotide is missing, RNA polymerase actually waits in the hope that it will come along. And then sometimes there are actually proteins that bind within the coding sequence to slow RNA polymerase down. It's not common, but it's known. So the answer to that is yes. Any other questions? Okay, let's go back to talking about transcription. We've almost finished. Okay, so we have here on one side of your screen a cartoon of a eukaryote, and on the other side, the cartoon of a bacterium. We don't say prokaryotes anymore, but this is an old slide. Okay, and what you'll see in the eukaryotes, what you know, is that the DNA is in the nucleus. It is transcribed in the nucleus. When it's transcribed, the introns shown here in yellow are removed, through splicing, and then the exons are joined together. Okay, and then a poly A tail is added, and a cap is also added. These help to orient the molecule. Okay, and then that molecule leaves the nucleus through a nuclear pore, goes to a ribosome not shown here, but they are in the cytoplasm, and then at the ribosome, there's a ribosomal binding site on the RNA. that is upstream of the start codon. This will allow the RNA to bind to the ribosome in the right reading frame. And then the ribosome will then, with the help of tRNAs, translate the mRNA to protein. Okay? In a bacterium, the process is much simpler and shorter. First of all, the DNA in the bacterium are suspended in the cytoplasm, although they're concentrated together in a nucleoid, but they're in the cytoplasm. Okay? And then the mRNA is produced by the DNA. There are no introns, so there's no splicing. It's just a quick, clean read. Okay? And then you get your mRNA, there's no capping and polyadenylating and so on. That mRNA goes straight to the ribosome to be translated. Did you know? I did not know this until I had actually years after my PhD. Did you know that in bacteria, it's possible to have an mRNA that's being made by transcription, and before the transcription finishes, it has already bound to a ribosome and translation has started. So the same molecule, the transcription is still underway, the other end of it is on a ribosome being translated. This is one of the reasons why bacteria have such a very fast reaction time. Because once transcription starts, a few seconds later, translation can start even though transcription is still happening. Whereas in a eukaryote, you need the mRNA to be completely transcribed, processed, introns removed, capped, tail, exported before you can start translation. So you always have a lag in eukaryotes that you will not necessarily see in bacteria. Okay? And then just to show you another thing I didn't know, when a particular gene is being transcribed, think about it. Sigma comes, sits down, and the other subunits start to transcribe. As soon as they start to move forward, it's actually possible and very common for another molecule of sigma and RNA polymerase to come right behind it. So before you finish transcribing one gene, another transcript is being written. This happens in both eukaryotes and bacteria. So one molecule is coming off, another molecule of RNA is coming off, another molecule of RNA is coming off, and so on. So when you look at these molecules of RNA, if you stain them, you can actually see that they look like a Christmas tree. Okay? This is the start of the gene, smallest molecule just coming off. This is the end of the gene where the larger RNA molecule is coming off. And you can see that in just a few seconds, we're making many molecules of RNA, each of which will go to a ribosome. So even though you have only one gene, just this Christmas tree here is going to produce, I don't know, about 30 molecules of RNA, each of which can then produce multiple molecules of a protein. So when we talk about scale up, the cell has been scaling up a long, long time ago. By the way, this is not a cartoon. This is a photograph. Okay, what was done here, the DNA was being actively transcribed, and then it was stretched out, and then the DNA and the RNA were fixed and stained, and then an electron micrograph was taken. So it's an actual photograph. This is what happens in the cell. Okay, so let's look at a gene. This is a cartoon of a gene. The gene from a eukaryote. This is the start codon, ATG. Okay, the next codon will be AAA. Okay, upstream of the gene will be a promoter. This promoter will never be this short. It will be typically at least 50 to 100 bases, and sometimes a promoter can be several kilobases. I'll show you an example in a minute. Okay, and then you can see that this gene, drawn as a cartoon on top, has got 1, 2, 3, 4 exons and 1, 2, 3 introns in orange. The introns are in the DNA. They're written in lowercase. Okay, so someone has looked at this sequence, figured out where the start is, where the stop is, where the introns are likely to be, and so on. And post-transcriptionally, there's a terminator somewhere there. Okay, so bioinformaticians do this a lot. We look at genes and then we figure out what the structure of the gene should look like, which is here. And then we figure out the function of the gene, and you're going to learn how to do that in bioinfo. Sometimes by comparing it to other genes of known function, sometimes by looking for motifs in the translated sequence of the amino acid. Okay. All right, I promised to show you a long promoter. So let's show you a fun one. This is one of my favorite promoters. It is the arabinose promoter in E. coli. It's the promoter that is upstream of the arabinose genes. And it's actually not a good promoter. It doesn't have good -10, -35 sites for RNA polymerase to bind. So it turns out that there's another promoter, another protein, not promoter, another protein, okay, which is called the arabinose C gene that binds to the promoter and does one of two things. If it binds in one conformation in the presence of arabinose, shown in orange, it recruits RNA polymerase, which allows the genes to be transcribed. The BAD genes are involved in metabolizing arabinose. So in the presence of arabinose, the ARA-C protein takes this shape and recruits RNA polymerase and allows the arabinose BAD genes to be transcribed. In the absence of arabinose, the structure of the ARA-C protein is different. And instead of binding right upstream of the other arabinose genes, it binds in this conformation. One molecule binds here and another one binds far, far away. And then it bends the DNA. Because the two molecules of RC, you see they always stick together. So if they're bound in this conformation, they're sticking together and allowing RNA polymerase to move forward. If they're bound in this conformation, in the absence of arabinose, RNA polymerase cannot access the promoter because they have bent the DNA, and then you cannot transcribe the DNA. So this is how E. coli manages to make sure that it transcribes the DNA to make the proteins necessary for metabolizing arabinose in the presence of arabinose. And in the absence of arabinose, it doesn't bother making those proteins because they're not needed. Okay. Any questions? Okay, a couple of reasons why I chose to show you the arabinose promoter here was because of ARA-C. I'm sure you can see from this picture, even though it's in 2 dimensions, that the shape of ARA-C and its ability to change shape in the presence or absence of arabinose are what dictates its function. Okay, that shape is written in the sequence of the amino acids that make the ARA-C and also in the nucleotides because they encode the amino acid. But once ARA-C comes out of the ribosome as a long strand, it's not going to be able to do this. It does this because it's going to fold in a particular conformation that allows it, number one, to dimerize. You can see dimerization is essential for ARA-C to function in either situation. And also allows it so that the parts that are going to bind DNA at particular sites, notice that the sites are labeled, that those parts are exposed on the right face of the protein. And also the parts that is going to recruit RNA polymerase also is able to do that. So it's a protein that can do many things based on its shape. And this is true of all proteins. So in your biochemistry classes, you will be taught about things that make shapes of proteins like alpha helices. You can see a helix here. This here is a protein amino acid sequence drawn as a ribbon so that we can see the types of folds that are in the protein. There's a helix here. Here, what we see instead of a helix is strands that are anti-parallel going like a zigzag. Okay, this is a beta sheet. So some bioinformaticians, not me, are more interested in looking at proteins than DNA. Okay, I'm interested in function. So I just want to know which protein is there. And so I look mostly at DNA and what it encodes. But there's a whole branch of bioinformatics that looks at the protein sequence and asks, how will it fold? And based on how it's folded, how will it function? Okay. This protein I'm showing you here is not RRC. This is a protein known as LexA, which binds DNA. And LexA is also a dimer with a flexible linker in between, and it binds particular sequences on DNA, just like this. This is a DNA below, and that's LexA on top. You can see that the exact structure of the DNA is very, very important. Oh, sorry, of the protein is very important for its binding. And LexA is actually something we refer to as a repressor. It actually binds DNA to prevent transcription. Okay, one of the things that LXA binds to in an E. coli cell is error-prone polymerases. Those polymerases I mentioned that don't have proofreading. LXA binds them, represses them, and turns them off. And the only thing that will make LXA let go is if LXA cleaves. You know, it's a homodimer, so that means two LXA molecules together. If those LXA molecules come apart, then they fall off the DNA. And they only come apart when DNA is damaged. So single-stranded damaged DNA floating in the cell causes LXA to fall off this, and then we tend to see the genes encoding error-prone polymerases transcribe because they're needed for repair. Okay? Unlike RNA polymerase sigma, LXA does not bind to a particular sequence, but it has its favorite nucleotides. So what's being shown here is a graph, 2 units up here. In position 17, LXA must always have a G. 16, it must always have an A. 15, it must always have a C. But in position 14, it likes to have an A, it can do with a C, and it can work with something else. Okay, so sometimes when we're talking about promoters or other parts of DNA where proteins can bind and the preferences are not 100%, it will be illustrated this way. Okay, I think this is where we are stopping. Yes, because we're not dissecting proteins. However, I hope I've given you a taster for the fact that there's a lot of bioinformatics that's done with protein sequence and protein structure. Okay, are there any questions on today or on earlier module 0? Earlier module 0 topics, or even things you've been reading on your own?
Okay, thank you very much, Prof. Please, does this mean that we all understood what Prof taught today and then from the previous sessions? So if you have any questions, just don't hesitate to ask them or drop them on the chat box, please. Yeah.
Okay. Good evening, Ma.
Good evening.
Yes, I have a question.
Mhm.
And my question is this: when you have a sequencing done you have sequencing done either by Sanger or NextGen, and at the beginning it tends to have some noise. We call them, I call it noise, and you have some ambiguous genes like the Y, S, M, R, and so.
Ambiguous nucleotides, yes.
Yes, ambiguous nucleotides, sorry. So those nucleotides, do they translate to protein? Can they be translated to proteins?
So the ambiguity that you're seeing is not in the organism that was sequenced. The ambiguity is in your data. The sequencer could not see the nucleotides. It doesn't mean that the nucleotides were not there. Does that make sense?
Yes.
So in the organism, you don't have Y and M and R and all these and N in the organism. In the organism, you have proper nucleotides. But your sequencer, for example, a Sanger sequencer starts sequencing from a primer, and at the beginning doesn't give you enough signal. So at the beginning you get N, and R and Y and so on. But that doesn't mean that in the organism that you were sequencing, the DNA sample, that you had N there.
Yes.
What it means is that there was a nucleotide there. It was either T, A, C, or G, but your sequencer did not see it. So the way you solve that problem is to start sequencing at another site ahead so that you can see what was in the place that you couldn't see. Okay?
Yes, ma'am. Okay, now the second question is this. If we are using, if we are targeting several gene regions, does the primer pick a particular point in the genome to start sequencing from?
A primer is so called because it was synthesized to match, to complement another sequence. Okay?
Yes.
So when you're sequencing with a primer, or when you're PCRing with a primer, or doing something else, primer extension, you're asking your primer to bind to complementary sequence. So you would have designed your primer based on what you want to do. Okay, if I want to sequence a particular gene, one way to do it is by Sanger sequencing. One way to do it is to make a primer that is complementary to the start of my gene. Now you will ask, well, what if I don't know the start of my gene? How do I then make the primer? What we sometimes do is we take the DNA we want to sequence and we bind it to DNA that we know. We use a ligase and join them. And then the known DNA is an adapter, and then we can put our primer in the adapter to sequence our sequence. In fact, this is another technique for getting away from the fact that from Sanger sequencing, the first few nucleotides will not be clear because the sequencer is not seeing them. You can put something ahead of the first few nucleotides so that what is not clear is what you know. And then by the time you start to get good data, you're into your sequence that you want to generate. So the primer is designed by the investigator for the purpose that the investigator wants to use it for. And the investigator would have decided, I want the primer to complement DNA at this particular position. Okay? When I talked about 16S ribosomal RNA sequencing, I mentioned that there are portions of the 16S ribosomal gene that are conserved, and we can actually use that for sequencing. For example, if I want to use the variable regions 3 and 4 to identify my organism, I can put my primer here because for all gram-negative organisms, this region here, the sequence is the same. So I can actually make my primer to match any gram-negative organism. I've gram-stained my organism is gram-negative. And I'm sure that my primer will let me sequence from here, which I know, to here, which I don't know. Okay?
Yes. Thank you, ma'am.
Okay, Bolatito has a question in the chat. I realize that there's a link between primers, sigma factor, promoter sequence, and then transcription. Please, can I break it down for better understanding? First of all, the link is today's lecture. They're all completely different things. I think if I'm going to look for what they have in common, they all involve binding DNA, but that's pretty much it. Okay? A primer. A primer is a piece of DNA or RNA that is complementary to another piece of DNA, which is a template. So a primer binds a template. Inside your cells, when DNA is being replicated, DNA polymerase can never start, it can never add a first nucleotide from nothing. Okay? It always needs to add a nucleotide to something else. Now, RNA polymerase, and if you think about it, RNA polymerase needs to be able to start from nothing because RNA polymerase's job is to transcribe. It starts at promoters at all sorts of places in the genome. It needs to be able to start from anything. Okay? So RNA polymerase can start, it can put a first nucleotide. DNA polymerase cannot. So when DNA is being replicated in a cell, your cell will first of all put a short sequence of RNA. That is called a primer. The name primer means to start. So RNA polymerase will put a short piece of RNA, and then DNA polymerase will then start adding nucleotides, the first one to the short piece of RNA, and then to other DNA. That's how your DNA is copied. Now, if I'm copying DNA in the lab, I don't want to buy RNA polymerase enzyme and then buy DNA polymerase enzyme. I just buy DNA polymerase enzyme and I give my reaction a primer that is complementary to the DNA that I want to copy, one end of it. I was giving this example. So my DNA will be complementary to this position. And then I'll put that sequence of DNA into my reaction. And then the DNA polymerase will then start adding nucleotides to the sequence that I put in. That's how PCR works. Okay, so primers are essentially short pieces of DNA that will allow DNA polymerase to add nucleotides. In the cell, they're used for replication. In the lab, they're used for PCR, which is replication. We also use them for sequencing if we're going to be synthesizing DNA as we sequence. Okay, that's what a primer is. A sigma factor is a part of RNA polymerase. RNA polymerase is the enzyme that copies DNA into RNA, is the enzyme that does transcription. The sigma factor is what helps the RNA polymerase to bind to the DNA that it's going to transcribe. The sequence that that sigma factor binds to is the promoter. So it's the sequence upstream of the gene that is going to be transcribed. Transcription is reading DNA into RNA. It's a form of copying because we're making the complementary sequence of the DNA in a different font, so to speak, in RNA. Okay? Whereas what the ribosome is going to do is not transcription, it's translation, because it's going to translate the language of nucleotides into the language of amino acids. Bolatitu, are we okay?
Yes, ma'am. Thank you, ma'am.
Okay, suggestion. Go to NCBI, get a nice textbook on molecular genetics. Read the chapter on DNA replication and read the chapter on transcription. I think one of the things, thank you for asking the question. One of the things the question is bringing out for all of us is that all these terms, I've dropped so many terms in three classes. Some of them, when I drop them, I'm careful to explain. Some of them, I forget that you don't know the language. You need to understand these terms. Otherwise, when bioinfo proper starts, your instructors will just be dropping them and it will sound like they're speaking some other language. So go and get your vocabulary straight by going over the module 0 background and also reading a basic textbook. Okay, very, very important. If you don't want to read a textbook, if you want to use something more visual and more interactive, there's an online resource called DNA from the Beginning. It has modules online, a little bit of history, a little bit of biochemistry, a little bit of genetics, how all these things I talked about worked. It's also, it was made several years ago, so it's not very data greedy. Okay, it doesn't have long videos and that kind of thing. So a little bit of data, you can go through the modules on DNA from the beginning. Okay. Any other questions? Okay, it's been nice having you. I recognize that you guys are all volunteering to learn. This is something that makes me very excited. You won't see very much of me in bioinfo unless the bioinfo team invite me. You'll see them. They're much more fun. And I really hope that your journey into bioinformatics and genomics is a fulfilling one. I believe that Tochukwu will have some announcements about bioinfo and how it's going to proceed going forward.
Okay, thank you very much, Prof, for the wonderful session so far from 2 weeks ago up to today. We really appreciate the time and the efforts you've made. So thank you very much. So going forward, the initial plan was to have the test next weekend, but due to the public holidays, for those that will be traveling home. So we'll have that by upper week, probably on Saturday, 14th of next month. So ahead of that, I encourage all of us to prepare very well for the tests, because that will be the determinants of proceeding to Model 1 for the main bioinformatics course. And believe in me, like if you are able to scale through that with zero tests, like what we have for you going forward is just very amazing. Like we are basically going to cover like more of kind of a full stack bioinformatics course for us across the 5-6 months for everybody. So do well to very very well for the test, and yeah, thank you very much. So I don't know if Abib has any other thing to add to that. Okay, someone has a question. Chidema, you can go ahead.
Yes, thank you very much. I just wanted to ask, like, when will the test dates be communicated, and can we like send feedback on it? Because I'm pretty sure I might also have a test next week Friday.
Okay, it's going to be on a Saturday, and that should be upper week, 14th of June precisely. So on a Saturday. So I don't know if that's going to clash with any of your schedules.
Yes, 14th of June, I'm traveling.
Okay, during that period.
Sorry. So I don't think Saturday will work, because we'll be using the faculty, and I'm not sure probably the faculty will open their auditorium on Saturday. So probably most likely it's going to be on Friday.
Hello, please, Abib, can you come again? I don't think I got you.
So I was saying I don't think probably the test is going to hold on Saturday, because the faculty won't, they won't be opening their auditorium on Saturday. So the only option that we have is on Friday, actually, if you want to make use of the auditorium, the faculty auditorium.
Okay, thank you very much. So I guess since we have 2 weeks, we would make arrangements and then communicate to you guys later on ahead of time. Yeah. Okay, you can go ahead, Adeola.
Hi, good evening. Please, what's the format of the test going to be? Is it like OBJ or theory or German, or like a combination of?
Okay, so it's going to be like a blend of subjective and theory. Yeah, but don't be afraid. I'm sure if you study well, you are going to pass. Yeah, just go with what Prof has taught across the past few weeks. Yeah. Kelechi, you can unmute and go ahead with your question.
Okay, I wanted to ask, since we are not writing tests next week, is there class next week? Is there a class next week?
Okay, so there will be no class until after the tests. Yeah.
Okay, thank you.
Yes, thank you. Is there any other question regarding the Moodle Zero tests or any other thing? Okay, David, you can go ahead. Yeah, David, you can go ahead with your question. I think your mic is still muted. Kindly unmute and go ahead with the question. Okay. Or any other person. Okay. Does that mean we are all good to go ahead of next week? Since we don't have any other questions. Okay. Abib, any other input from you?
Oh, no. I have anything to add?
Okay. And Toby is not here, and Anderson is not here too. So, but nevertheless, if you have any other inquiries you need to make, you can just simply email us, and then we'll get back to you. So thank you very much, everybody, for joining. Thank you, Prof, for the time. We really appreciate. Yeah. So thank you, and bye. Good night.